Very warm welcome to our biology class. In this wonderful class, we are going to learn regarding some structures present on the bacteria. Bacteria are classified into two categories based on whether they are motile or non-motile. Motile bacteria are called as trichous bacteria and non-motile bacteria are called as atrichous bacteria. Non-motile bacteria do not have any structures on the surface of them for the motility, but the motile bacteria have a flagella. Some bacteria have single flagella, some other have number of flagella. Based on the arrangement and number of flagella, we classify the motile bacteria into different categories. Some bacteria have single flagella on one side. Such bacteria are called as monotrichous bacteria, and certain other bacteria have two flagellas on both the sides. Such bacteria are called as amphitrichous bacteria, and certain other bacteria have tuft of flagellas, means the group of flagellas present on one side. Such bacteria are called as lopotrichous bacteria, and certain other bacteria have two group of flagellas on the either side. Such bacteria are called as amphilopotrochus bacteria, and certain other bacteria are called as peritrichous bacteria. These are have flagellas on the entire surface. Now let us get into the details of different parts of flagella, how it is arranged on the surface of bacteria. Students, we already learned in the previous class about the different layers present on the bacteria. Bacteria have a plasma membrane. Outside to the plasma membrane, there is a cell wall, and outside to the cell wall, we used to find the presence of glycocalyx layer. And in addition to that, we have seen even the mesosomes. Bacteria have a flagella which is made up of basal body, hook, and as well as filament. Let us see that how all those three parts are arranged. Basal body made up of the different rings, and those rings are connected with the help of a rod at the center. So those rings are called as M ring and S ring. Both these rings are present on the either side of a plasma membrane, and one more ring called P ring present in the cell wall, and there is one more L ring present in the glycocalyx layer. All these four rings together, we call it as a basal body. And in gram-negative bacteria, we used to find the presence of only just two rings are there. Only M and S ring will be present in case of gram-negative bacteria, and gram-positive bacteria that have all these four rings. That is the difference between the gram-positive and gram-negative bacteria with respect to basal body. The rod present at the center of all these rings in the basal body is connected to the small hook-like structure. It is the second one more part of flagella, and the hook is further connected with a long filament. This is what the way how the flagella is made up of in prokaryotes. There is a different structural difference between the eukaryotic and prokaryotic flagella. If you take a cross section of filament, we used to find the presence of nine microtubules located on the periphery. This is the 1, 2, 3, 4, 5, 6, 7, 8, and 9. So hence what we usually refer it as 9 plus 0 array. But in case of eukaryotes, flagella made up of 9 plus 2 array. It means at the center, two more central filaments are located. In addition to this flagella, there are certain other structures present on the surface of bacteria. Those are called as Pili and Fimbriraes. Let us even understand what are those structures and what is their function. We already understood regarding the filament. Now, we need to know that certain bacteria have extra genomic DNA, that DNA called as plasmids. We are going to learn regarding the plasmid in detail. So it is one of the most important vector which is well utilized in biotechnology field. So these plasmids have three genes. One is F gene and col gene as well as R gene. F gene is refers for fertility gene. So this gene is responsible for making the some surface outgrowths. They are called as pillies, and even these are called as sex pillies. So these are made up of the special protein called pillin, and it will help in forming cytoplasmic bridge between the two bacteria, and even it is going to help in conjugation. The bacteria, those who have plasmids and they are able to produce the pillies, they are called as F plus bacteria. It means the fertility bacteria. Even these bacteria are called as donor bacteria, or even they are called as an male bacteria. So at the time of conjugation, one more bacteria comes very near to each other, and the pillies are going to form the cytoplasmic bridge between adjacent bacteria. By that time, the extra genomic DNA, plasmid, or the genome from the DNA, host DNA, it is going to be transmitted into the other bacteria. It is called as F minus bacteria or recipient bacteria, or even we call it as a female bacteria. What happens by this time? The gene is going to be moved through the pillies, and once it is entered into the other bacteria, it will replicate and forms the one more plasmid. By this time, the second bacteria is even become an F plus bacteria. Even it can able to produce the pillies, and it can able to transmit its gene to the other bacteria. In addition to this, bacteria are even have some other external structures. Those are called as fimbriaes. Fimbriaes are comparatively smaller than the pillies, and these are helps in adhering to the other bacteria, or even they are going to help in adhering to the host cell at the time of infection, or they are going to help in adhering to the substratum. Suppose, for example, if the bacteria is surviving in running stream, then it need to be found attached to the rock at the bottom, and by that time the fimbriaes are going to play a very important role. I hope so, students. By this one of the short video, you all comes to know regarding the Flagella, Pili, and the fimbriaes present on the surface of prokaryotic cells. Thank you. Please share the video, like it, and comment on it. Thank you so much.